Hello students, good morning. Today we are going to continue our discussion on the search of genetic material. So Griffith was not only the one who performed experiments to find out which was the genetic material. However, he was the first one who performed the experiments using the S strain and the R strain of pneumococcus bacteria. However, there was another scientist named Hershey and Chase. They performed their experiments using bacteriophages to find out the genetic material. So here we will see this experiment will again prove the same thing, that it is the DNA which carries the genetic material, which maintains the genetic material. The experiment with bacteriophages. Now, before we go ahead with the experiment, let us quickly remind you what are bacteriophages. The name sounds similar to bacteria, but they are not bacteria. Instead, they are viruses that infect bacteria. As I said, these are the viruses that infect bacteria and they also replicate inside bacteria. So here is the structure which you can see. This describes the structure of bacteriophages. The structure broadly has three important parts. As far as the structure of bacteriophages is concerned, there are three broadly important parts of the bacteriophages. Let us quickly look at the important parts. So here you have a protein covering. So just here, this is the protein covering. So when we talk about the protein covering, so this is the protein covering, right? So this covering is made up of protein. So this is a protein coat. Inside this is the genetic material. This structure which you see is the DNA, and the outer coat is the protein coat. Understood? And here you see some structure at the bottom are the tail, as they are called as a tail, right? Now what happened is, how does it infect the bacterium? Now it fixes on the body of the bacterium. Gradually its tail penetrates the bacterium, and that is how the informations get into the bacterium and replication happens. Now basically it happens once the genetic material reach inside the bacteria. The bacterial cell starts treating this genetic material as its own genetic material. However, it is the genetic material of the virus, right? But since the bacteria start treating it as its own genetic material, so it will start manufacturing more viral particles. And that is how replication of this virus will take place inside the body of the bacteria. Now the question is, what is being injected? Children, as I said, it is the genetic material that is being injected. So what is the genetic material? Is it the protein that is getting injected, or is it the DNA that is getting injected? Because in this case, these are the two options that can be injected inside the body of bacteria. Because two things we have discussed: the outer protective covering that is made up of by the protein, and the internal stuff that is the DNA. So we have two options that can be injected inside the body of bacteria, and whatever is getting injected, that particle has the genetic information. That is why it is able to produce more and more viruses, right? So that is what we have to see here. This is how a normal bacteriophages react when it comes in the contact of the bacteria. So what was done in the Hershey-Chase experiment? Okay, now let us see what was done in Hershey-Chase experiment. Like Some bacteriophages were grown in radioactive phosphorus medium. So it was grown in a medium which has radioactive phosphorus. Now phosphorus is an important component. It is the important constituent of the DNA. We have studied. So if you look at the structure of DNA, you will see that the phosphorus, that is phosphate, is present, right, in each nucleotide. So phosphorus is an important constituent. So if the bacteriophages are grown in radioactive phosphorus medium, right, so it is definite that the bacteriophages will have be a radioactive DNA. अगर रेडियोएक्टिव फॉस्फोरस मीडियम में ग्रो करते हैं तो इट इज़ ऑब्वियस कि उसका जो डीएनए होगा वो भी रेडियोएक्टिव डीएनए हो जाएगा. दैट द डीएनए विल बी ऑल्सो रेडियोएक्टिव, बट द प्रोटीन विल नॉट बी रेडियोएक्टिव, राइट. व्हाई इट इज हैपन? रेडियोएक्टिव फॉस्फोरस में ग्रो करने पर, लाइक दिस इज द रेडियोएक्टिव फॉस्फोरस मीडियम, जब इसमें हम बैक्टीरियोफेजेस ग्रो करते हैं तो डीएनए कंटेंट भी रेडियोएक्टिव हो जाता है. इट इज शोन विद द रेड कलर. द डीएनए, रेडियोएक्टिव डीएनए इज शोन इन द रेड कलर, बट द प्रोटीन कोट will not be radioactive. Do you know why? Yes, exactly, because protein does not contain phosphorus. So obvious this portion will be the radioactive. So आप जो DNA content दिख रहा है रेड कलर से वो radioactive होगा, but the outer protein coat which is in the black color will not be radioactive for these bacteriophages. So let us denote like this. This radioactive DNA is denoted in the red color, right? So this denotes radioactive DNA, right? So this red color denotes radioactive DNA. So now there is another set of bacteriophages which were grown in the medium of radioactive sulfur, right? उसी तरीके से कुछ bacteriophages को radioactive sulfur में grow किया गया. Some bacteriophages grown in the radioactive sulfur medium. Now bacteriophages will radioactive protein. अब जिन bacteriophages को radioactive sulfur में grow किया गया, उनकी जो proteins थे वो radioactive protein हो गए, जो यहाँ पे आपको red color से outer covering दिखाई गई है. Now sulfur is the important constituent of protein, we all know, and sulfur is not present in DNA. Therefore, the bacteriophages which were grown in the radioactive sulfur medium, they will have radioactive protein, right? So here in this picture you see the outer coat is all red color, right? So the outer coat is all red color because now outer coat is radioactive in nature, right? It is made up of by radioactive protein, so that is why it is denoted in red color. So please remember, wherever you have red color, that is the radioactive, and the black color, right, that is the normal one. So these two type of bacteriophages were first grown, right? Now one is with radioactive sulfur and other one is with radioactive phosphorus. Where we have radioactive sulfur, so it has radioactive protein. Where we have radioactive phosphorus, it has radioactive DNA. And then what was done? So in case 1, the bacteriophages with radioactive DNA attached to the bacteria. Now as soon as bacteriophages come in contact of bacteria, the same process will happen. The tail will penetrate inside, right? So the bacteriophages come in the contact of the bacteria. So the bacteria will get infected. Now what will happen? The first step, infection is done. So 3 simple steps will take place. First is landing, second one is thinning, and third one is penetrating. After these 3 steps, the first infection process has been done. Now what was then observed? So what was the purpose to separate phage particle and the bacterial cell? We separated them in order to see which of them is radioactive, right? phage particle or the bacterial cell are radioactive, क्योंकि phage particle infect करते हैं bacteria को by transferring their genetic material, right? So now what was final result? Result was that radioactive bacterial cell, but no radioactivity detected into supernatant. So phage particle did not have any radioactivity in them, right? So in the case, what was radioactive? The DNA was radioactive. So this proves that DNA is passed on in the bacterial cell, right? So children, as I just mentioned, there are 3 steps by which a bacteriophages will infect the bacteria. So first, it was land on the bacteria, right? So here it has landed. Then it pins itself, so it get fixed on the surface of bacteria. Now it is get fixed on the surface of bacteria, and then its tail start to penetrate inside. So the tail of the bacteriophages start penetrating itself into the bacteria. So this is how it will infect the bacteria, right? So what happens now? So now once it infects the bacteria, then inside the bacteria the genetic material is now present. So here you see the red color DNA in the bacteria. That because the genetic material has got transferred into the bacteria, right? So now these bacterial cell, along with the genetic material which has been passed on by bacteriophages, this entire thing is agitate in the blender. So the next step is what? It is the blending. The next process is the blending. Now what will क्या देखा हमने? That the genetic material is passed into the bacterial cell. Now this entire thing is agitate in the blender, and centrifugation lead the phage particle as supernatant, और जो phase particle होता है वो as a supernatant find हुआ. Now does this happen? Now when centrifugation takes place, what happen? The particle with different densities, they get separated. Okay, so the particle which has heavier, they tend to come nearer, they tend to settle down at the bottom. Okay, and the lighter particles tend to come upwards. Okay, so that is what happens in the centrifugation. यही तो होता है. Centrifugation is the separating technique. So in that way, the particles with the heavier density, they settle down at the bottom, and the particles with lighter density, they actually come upward, right? Now when this underwent centrifugation, it was found that the phage particle are present supernatant, right? They are as the supernatant, they are present in the supernatant. So it was observed as supernatant is the liquid which are present above the residue. So the bottom is the bacterial cell. Like children, let us suppose this is the container in which the centrifugation is done. Okay, and when centrifugation is done in the blender, then what will happen? Some of the content is settled down with the heavy densities, and some of the substances come upward with the lighter density. Now here what is supernatant? It was observed that supernatant is the liquid which are present above the residue. So this is the supernatant and this is the residue, right? So the bottom is the bacterial cell. This bottom is what? It is the bacterial cell. And above were the phase particle, और जो supernatant है वो क्या है? Phase particle है. So this blue region is the phase particle जो कि supernatant है, जो कि fluid है except the residue, और जो residue है it is present at the bottom. Got it? Now let us try the same thing with second set of bacteriophages with radioactive protein. अब रेडियो एक्टिव प्रोटीन इस सेकेंड सिनेरियो की अगर जब हम बात करते हैं, what happened in this case? Similar things, right? First the bacteria was injected again, it is agitated in the blender so that phage particle can be separated from the bacterial cell. Centrifugation की process हुई और bacterial cell phage particle से separate हो गया, right? And because of the different densities they are separated out. But in this case it was observed as the result that there is no radioactive bacterial cell. जो residue था उसमें radioactivity present नहीं थी, but the radioactivity is detected in the supernatant, और जो radioactivity थी वो supernatant में present थी. That is radioactivity is present in the bacteriophages but no radioactivity in the bacteria, right? That means in this case radioactivity is not transferred from the bacteriophages. इस केस में क्या हुआ, radioactivity transfer नहीं हुई, so it has been not passed on. What is not passed on? Radioactivity with proteins, right? So that protein is not passed on. It is not a genetic material. So in that way it is suggested that protein is not a genetic material. पर पहले केस में क्या था, वहाँ DNA था. Phosphorus is a part of DNA. It is the constituent of DNA. And with that constituent, the radioactivity is passed on into the bacteria. So that means DNA is the one which actually transferred from one part to the another part, from the bacteriophages inside the bacteria, right? So in that way it is confirmed that the genetic material is DNA. So by this experiment, Hershey and Chase experiment, it was suggested that DNA is the genetic material. This experiment of Hershey and Chase in 1952, they proved that the genetic material is the DNA. Got it? So very soon we will meet again. Till then, have a nice day and thank you.